Hi, my name is Esther Tok, and this summer I was working on characterizing the diversity and life cycle dynamics of chytrid fungi. So you might have heard of this pathogen completely sweeping across the world and killing amphibians all over. These are actually chytrids, which are unicellular microorganisms with zoospores that can either crawl or swim, similar to sperm, or also grow into these sporangia that can mature and release more zoospores. These chytrids have huge ecosystem impacts, just like the pathogenic BD, which is the one killing amphibians. And pathogenic chytrids, this means that they can infect and cause disease in their hosts. There's also saprotrophic chytrids, which consume detritus, like plant waste and animal waste. So these ecosystem impacts really interested me, and I wanted to better understand um, the different types of chytrids we had at our local Jasper Ridge Biological Preserve. I hypothesized that there were going to be chytrids there due to previous sampling efforts for just BD, and also I predicted that the biodiversity would vary between the different bodies of water we sampled at, since the lake is more stagnant than the creek. After we got these water samples, we brought them back to the lab and we added white onion skin, pollen, and chitin, which are three different baits that have traditionally been used by other chytrid scientists. We saw that the canonical baits prepared in this method were not very effective for baiting chytrids. We think that there's three different possibilities as to why this is. There's either first, not many chytrids at Jasper Ridge. Second, the chytrids just didn't like the bait preparation method that we used, but they did like the compounds themselves, or the Jasper Ridge chytrids just didn't care for those compounds to begin with. But still, we did find chytrids and also organisms similar to chytrids. This is another zoosporic fungi. And after this, we plan to purify and isolate them and identify the different species using morphological analysis, which is using the different characteristics of them, like their size, their shapes, to figure out what species they are. We also could possibly use molecular analysis, which is using PCR to amplify and sequence a specific DNA fragment and comparing it to known sequences to figure out the species. So thinking about pathogenic chytrids, specifically BD, we wanted to better understand how concentration can impact its life cycle timing. An infection zoospore density could really impact how quickly infection spreads if zoospore density is impacting how fast they grow and release more spores. So we did this experiment by first isolating the zoospores, and then plating them at different concentrations, low, medium, and high, and then measuring the diameter of those cells using a software under the microscope. So we did do a lot of optimization for a bunch of different conditions for the microscope, and eventually we were able to figure out a way to get those time lapses to record pretty consistently and robustly. However, we were still only able to get in-focus time-lapse from one plate, the high-density plate, and we were able to generate this graph. So over here, this is diameter plotted against time, and you can see over time, sporangia size increases and then plateaus as the sporangia begin to pop and release their zoospores. In the future, I want to try to get comparable results by taking these time lapses on a high throughput microscope with autofocus so I can actually get comparable data. And with this comparable data, afterwards, we can conduct additional experiments to better understand how and why concentration impacts chytrid life cycles, to give us more insight on how infections occur in amphibians. So in all, understanding chytrid diversity and also their life cycles was able to give me a better understanding of the organisms that shape and inhabit our surroundings. Yeah, thank you very much.